Lovely to have some of you here in the lecture theatre with us. We have two very special guests. We have Nick, Rachel's husband, and Ruth, her sister. I'm going to be allowed to take this off for a moment just while I'm speaking to you, but you perhaps have a chance to chat in the bar. And when you speak to them, Ruth, you'll understand where their childhood was. Your lovely Scottish fur, Rachel, and your education in Edinburgh, which I was very interested to learn about. I won't steal your thunder and take all your slides and tell everybody what's happening, but what a pedigree. If you look at illustrious institutions, Edinburgh, Oxford, Cambridge, and of course, St George's. So what a stellar career. It's lovely to have you here. We now would like you and require you please to keep your masks on if you're in the audience, but Rachel has permission to remove hers, so it's easier to understand her. Not that I've ever had any difficulty understanding Rachel, but just from the point of view of the recording. And as always with inaugural lectures, she has the last word. Nobody's allowed to quiz her and give her cross-questioning at this juncture, but there's plenty of opportunity to do that in the bar afterwards. So please do come upstairs and enjoy the celebrations with us. After Rachel's finished talking, Jodie is going to come forward and give the vote of thanks. So thank you very much for that, Jodie. But I have to say I couldn't miss the opportunity of your inaugural, Rachel, to say a huge personal thanks to you. I think you're an absolutely fabulous colleague. I highlighted some of your many positive characteristics, your clarity, your intelligence, your ability to tell me, quite frankly, the truth, which I've so valued. You were one of the people I met fairly early on when I came here to George's. And when you arrive new in an institution, you hear whispers about people. Lots of little bit of background. Everybody wants to give you their 5 pence worth on who it is you're about to meet. And I have to say that universally, the feedback on you was incredibly positive. They said, oh, she's very good. She gets things done, that Rachel. And boy, don't you get things done. So I had a really good preamble to meeting you. But also, as soon as I did meet you, it was quite clear to me that you were somebody who was very important to the institution and somebody who was going to make contributions over and above. And so you have done. So it's a great, I want to say thank you. We're lucky to have you and we're all looking forward to your inaugural lecture. So thank you very much.
So, hello everyone. Hopefully you all know who I am, but I thought I might just start by explaining the title because I'm going to talk a bit about things that I've done, and it'll be a science part at the beginning and talking about my studies from my PhD onwards and uncovering new roles and indeed new forms of what have been thought to be very well characterised molecules. But also I'll talk through, my time at St George's and some roles that I've taken on, but also, with the purpose of those roles being to help other people in their roles and development, both in research and, for example, in the NHS and social care. So just in case I lose track when I'm lecturing, that's what the general theme is as we go through. In terms of the early days, I was born in Kirkcaldy in Fife in Scotland. There's the family. You can probably guess which one's me and which one's Ruth. And that's us in our school days. Sorry, Ruth, I'm going to point out the blue Peter badge that people can ask you about later. And actually, if you look just in the top of that one, you'll see a little bit of grey. That's the Victoria Hospital Kirkcaldy, which is actually where I had my first job, which was in the pathology labs, where I would carry bits up the stairs and watch as the pathologists dissected them. So there's a bit of background there as well. I went to Bulweary High School, which used to be the local comprehensive school in Kirkcaldy. And when I was there, I think in terms of thinking about the future, it was more that I knew what I didn't want to do, which was medicine. Sorry to most of the audience there and my sister, but I knew that was what I didn't want to do. But I'd done the standard biology, chemistry, physics, maths, English, and I think it's a marker of how useful university open days are, that I went to one, sat down at a table with somebody from Edinburgh University, and they said, well, what do you want to do if you come to Edinburgh? And I said, I don't know. And they said, what hires are you doing? And I told them, and they said, well, what you want to do is biological sciences, because actually if you do that, you can go into lots of different honours schools. You can go into all the biological sciences ones. You can also transfer to chemistry, or you can transfer to psychology. So if you don't know what to do biological sciences. And that's what I did. So I went to Edinburgh University. This was my lovely Halls of Residence, which is Milnes Court. It's still part of the Halls of Residence at Edinburgh University. And you can see it doesn't look like your traditional halls of residence. It's actually at the top of the Royal Mile in one of the little courtyards. And so, you know, depending on the time of year, we'd hear the Edinburgh Tattoo going on. There would always be tourists going by, and it was a fantastic place to live. But actually, in terms of studies, I was out at King's Buildings, which was the science campus there, which was about, well, by my walking was an hour. Possibly other people could do it faster than that, or a bus journey away. And as I went through university, and I did really get a real joy out of the studies there, and a real interest, partly triggered by a book called Jurassic Park, and partly by some people that we taught. Molecular biology was the thing that really appealed to me, studying DNA and its manipulation and what you can do with it. And, you know, an absolute giant at Edinburgh University was Ken Murray, but also his wife, Noreen. You can never have had one without the other. And Ken was my BSc project supervisor, and you know, he was really inspirational to me because Noreen, did a lot of studies of restriction enzymes and how you manipulate DNA. Ken was very translational, and so he set up a company called Biogen, and he also came up with the first recombinant vaccine for hepatitis B virus, and actually through the patent for that gave a lot of money to Edinburgh University, which I think, as we were told, at least as we were told, funded that building there, which was the molecular biology thing. And Ken was my BSc project supervisor. His lab meetings were quite an experience. I was not the most studious student, and that didn't pass muster with Ken. He really pushed people. He pushed, and you know, at the time when I was coming in my final year to thinking about what to do next, I spent a lot of time doing music, and while I was at Edinburgh, I played the saxophone. I took the grade 8 exam. My music teacher was, you know, strongly saying you should think about going and doing this, and I was thinking about a postgraduate course at the Guildhall School of Music in Jazz, which was the particular one if you're in that area. But I was also thinking perhaps of a PhD, and it was Ken who really pushed me on that. And you know, he said, well, you know, you've got to start at the top. You start Oxford, Cambridge, and then everywhere else, and you wait till you've been turned down by one before you go on to the next. And you know, I was, you know, a bit unsure about that, and I did apply to all these universities, and I was offered an interview at Oxford, but I was thinking, do people like me go to Oxford? And Ken, being Ken, basically got his secretary to book me a train ticket because he wasn't sure I would go if I did. And he said, just go, just meet them, see what you think. You're interviewing them too. And I did, and I loved it when I went there, and so that's where I ended up. So that's why Ken was particularly key for me. And so I ended up doing a DPhil at the Institute of Molecular Medicine in Oxford, in Andrew McMichael's lab, but also with Paul Burness was my other supervisor. Paul also was a jazz musician, so that came in very handy too, as I went through the PhD. And Andrew's lab focused on T-cell recognition of HLA class 1 proteins. This is where immunology goes into the letters and numbers, and people start losing track. And certainly one of my fears, I've not really done immunology at university. There'd been some lectures, it went over my head. So immunology I thought was not for me. And then I ended up doing a PhD in an immunology lab, but it just happened that they were looking for somebody who knew about molecular biology to do lots of DNA sequencing. So that's what the lab focused on, mainly on HIV. That was the main focus of Andrew's lab, but that wasn't what my PhD project was on. Mine was on a particular variant of HLA class 1 called B27. So what are HLA class 1 proteins and what they do? If you're not an immunologist, what I would say is to think viruses infect cells, they go inside cells and they multiply there. But how does your immune system know what's going on inside a cell? So this image is a slice through a cell with its nucleus there, and the green bits are viral proteins. But how could a T cell, whose intention is to kill any affected cell, actually see inside a cell to know what's going on? Well, that's where these HLA proteins come in. And probably the thing I spent most time on for this lecture was thinking of a way, how could I possibly, to a non-immunology audience, explain some kind of, indication of what HLA class 1 proteins do on the cell surface, and it was the macaron picture that I came up with. So if you're an immunologist, look at the left side. What HLA class 1 do is they take up a chopped up sample of all the proteins that are being made inside a cell. So they take a sample, they carry it up to the cell surface, and it's that little red bit up there, and they hold it up at the cell surface for the immune system to scan. And by scanning these at the cell surface, the immune system gets an idea of what's going on actually inside that cell. Or if you want the macaron analogy, on your cells are covered like this with HLA class 1. You've each got 6 different genes, and it's the most diverse part of the human genome. They're really variable, so you'll each have your own 6. It'd be unlikely that any 2 people in this room, unless they're twins, have the same 6. There are hundreds of variants, but you only have 6. So you have these, and they each have a filling. The filling will only work with particular flavors, so it's got to have the right filling being held up by the protein. And you can think of the T cell perhaps like a very fussy elephant that comes in and snuffles over all of these with its T cell receptor. And it looks at 2 things. It looks at the biscuit to say, is this one of mine? Should this be there? Is this what my genome encodes? And also, is the filling the right one? And is it made from something that's normally in the cell? If the answer to either of those is no, it kills the cell. So I said there were hundreds of variants. It's the most variable part of the human genome, but that also means there's disease associations with different variations, and there are plenty of diseases that show disease associations with particular, say, flavors of macaron or particular variants of HLA. The most striking one is ankylosing spondylitis, which is an arthritic disease. It's associated with HLA B27, and that's what my PhD focused on, was to look at this relationship and see why it might be that this particular variant, B27, causes disease. Now, that disease association was first published, I think, in 1972 or 1973. And I started my PhD in 1993. So, it was well established by then. Bit of a stretch to say, can you discover, what's going on here? But certainly working towards that would be helpful. So I think I was one of those PhD students who went through multiple projects, largely because we tried one thing, didn't really get results, went on to the next. And because the role of these HLA proteins is to go to the cell surface, present an intracellular sample there, which is then seen by a T cell, The thinking in the field very much at that point in time was it's got to be something to do with the T cell and its receptor, or it's got to be something to do with the peptide, the filling that's being presented. And I had projects that corresponded to chapters looking at the T cell receptor. Was there anything special about the T cell receptor that some of these? No, that was a year gone. Tried predicting peptides to see if they might trigger cross-reactive responses to cell proteins. No. Tried purifying peptides from the protein that had been normally processed in cells. No. And one thing that was particularly frustrating is that although it wasn't a mutated gene, this B27, any kind of assay that you put it in, it never behaved like all the other HLA class 1. You know, the assay that we used here, you would normally get some cells, lyse them, the MHC would fall apart and you could purify the peptides. If it was B27, you'd have to do that, boil what you got for 5 minutes and then purify the peptides. It never worked like the rest. But you know, these are all projects, and I think this added up to about 3 years of getting things I could write up, but meh, you know, who cared? To some extent was my feeling, certainly as a depressive PhD student, when you reach that point, and you're thinking, well, you know, what's this? And then my supervisors, you know, suggested that I tried refolding B27, making it in bacteria and refolding it, not for my own project, but actually for the people who were studying HIV in the lab. To do that, fluorescently label it, and then the HIV people could use it in their studies. Needless to say, B27 did not behave like other MHC class 1 when you tried to refold it, and I don't know how many months, almost a year of trying to refold it, and it never worked. I ended up having to mutate it to get it to work. But with the unmutated form, I'd just been looking at things, and this was kind of like the seminal experiment, as it turned out, for my PhD, which is quite low tech now. But we would try refolding, and then you'd run it through this tall chromatography column. Anything big, usually misfolded protein, would come out first. So that would be this big peak here. And then if you had refolded protein, it would come out later. And one day I was lazy and couldn't be bothered to change the column from one that someone had else been using. It wasn't the usual column. It had much better separation of larger complexes. And I saw a peak there, which was equivalent in size to a dimer of just part of the HLA protein. It was forming dimers with itself through a cysteine disulfide bridge. And Andrew and Paul were great supervisors. They said, hey, why don't you just go on and look at this and see, is this something interesting? And I always think of it as a kind of Jekyll and Hyde thing. So going back to the macarons, you know, if most of your B27 in the cell is normal, but some of it is this alternatively folded form, what does that mean? And you know, what might the results of that be? And I won't go into it here, but you know, I folded it, studied it, purified it, found that these dimers could still bind peptide, still had structural integrity, but they weren't seen by the T cell receptor. And so I, you know, was there for not beyond the usual time for a PhD, but it really took off really in the last year or so with that work. And it's something that's still to be studied in the ankylosing spondylitis field, is these alternatively folded forms. And there are differing opinions as to whether they're important, and if they are important, what it is that's going on in them. But then I went on to a postdoctoral post at Cambridge University, which was in John Trousdale's lab. That's John there, another person who was a jazz musician and jazz fan, so that continued that theme for me. And this was the Department of Pathology there where I worked. The lab I was in was that one up there. And what John's lab had been doing for some years was sequencing a complex of relatively, at that time, uncharacterized genes, which recognized MHC class I proteins. And so they had lots of reagents, lots of things, and I could then take this alternative form that I'd found in my PhD, and in John's lab, go on to see, do any of these other receptors recognize it? I probably, from John's point of view, spent too much time doing this when I was in his lab. I got press ganged by a PhD student in the lab because of my height. Why don't you take up coxing? And really loved doing it. I think I was never a competitive person, particularly, until this turned me into a bit of a monster in terms of, I used to cox men's crews, and being the only person who could speak for 2 hours can be terrifying when you first do it, but is really enjoyable, you know, and they have to do what you tell them. And, you know, I loved doing that. I think it gave me a lot of confidence, but, you know, it also gave me that, you know, before I'd always been a bit, oh, I don't know what I'll do. And that after that, I would kind of be the person that once I decided to do something, I really went for it. And so it had quite an impact on me. But anyway, back to the science. So I mentioned that I had my alternatively folded form of HLA-B27, which I called HCB27 and published with that name, but then nobody else stuck with it, so you won't really find that in literature now anymore. But made it, and then made these big complexes of it with fluorescent labels. And what those do is you can effectively use them like an antibody to stick to cells and see what they're sticking to, and that tells you that it's being recognized by those cells. And with all the reagents in John's lab, I found there were these receptors, which are called LILR, and some of them could bind both normally folded B27, and anything above that line means it's binding, but also the alternatively folded form. And from then on, really, my focus was more on the receptors, because everything that we kind of traditionally thought in immunology about HLA proteins was they're important in telling the difference between self, which is good, non-self, bad, kill it. These receptors were seeing that protein, but they weren't on killing immune cells. They were on monocytes. And what they do, I always think of them as like volume controls of the immune system. They could either amplify or inhibit effectively the functions of those cells. And that was something that I continued to study and work on. And I did various studies just looking to see what these receptors, these LILR, do in different cells and what happens when you trigger them and so on. And that's what took me a bit further on. In fact, until I came to St. George's and set up here. And partly through some work with some people who got in touch from Boston, Harvard University, Jim Yu and Matthias Lichterfeld, and some of their observations in their studies of HIV patients. We started to think a bit more, and this was really led by Des Jones, who was a postdoc in the lab. And we were thinking, okay, these LILR, they don't distinguish. They're not specific like T cells are. They just see everything if it's an HLA. So, you know, how does that work and what's the point? And we started to think, well, they might bind to every different HLA, but is the strength of that interaction the same for all the hundreds of different variants? And so Des came up with this great study using reagents that were actually used for transplant tissue typing, but looking to see, did these LILR receptors stick with the same intensity to all the different variants of HLA? And the answer was no. For one particularly inhibitory LILR, we saw some quite notable variations in binding, particularly for HLA-B27. Interestingly, we find that the activating, that turn the volume up on the immune system ones, quite like unfolded HLA class I. So that work is still going on in the background for various people who work in the field, that they seem to be seen by activating receptors on natural killer cells as well as monocytes, the alternatively folded forms, which it turns out now are not just B27, there's some other HLA that do that too. But we thought this was quite an interesting result because we were seeing different strengths of binding, which led to the next question: does different strengths of binding have any physical effect in terms of immune functions? And that was where Zhu and Matthias came in again, and wanted to look, at disease associations of different HLA class 1 alleles. And I talked about ankylosing spondylitis, but that's quite an extreme one. HIV is another disease. This is a genome-wide association study of different genes in HIV infection, their impact, and that there, the blue line sticking up, it's called a Manhattan plot, corresponds to where the HLA genes are. So that there is an important role for HLA variation in terms of how people respond to HIV infection. People who tend to go years without ever progressing to AIDS tend or often have particular HLA variants, whereas people who progress very quickly often have other HLA variants. So Zhu and Matthias took the data that we had from this study of DES and fit it in, with some particular cohorts of HIV, big cohorts of HIV patients, and with a lot of very complicated statistics. And I've left the slide out because I don't understand all the statistics. But what actually turned out was looking at all the 6 HLA that people had and the strength of their binding to the receptors correlated with how much virus was circulating in their blood, how well they could control infection. And what we found was that weak binding to this particular receptor, which turns down the immune response, correlates with effectively good outcomes with low viral load. So the weaker the binding to the turn it down button means there's less turning it down, there's less inhibition of immune responses, which correlated with better control of infection. And from that, Laura, a PhD student in my lab, and I came up with what we thought was a new model for disease associations for these proteins. We tried to put that in the title. We called it a new model for disease associations, and reviewer one said, unless you can prove it's a new model, you have to take the word new out. And then we had this whole debate, is how can you prove it's new? All you can do is see that you can't find anyone else has done it. But anyway, we just took it out. But for me, I love this idea of, I think HLA in disease can be T cell, can be natural killer cell, can be monocytic cells. And if anyone wants to talk to Alex, he's now working on these now, particularly around bacterial infection. But they're powerful proteins. They really do have quite an impact. And more we're seeing in the field, you're seeing them coming up in cancer studies where you see the inhibitory ones. During acute infection, you see some of the activating ones, but also balancing factors in the immune response. They do seem to be quite important receptors. And I do think it's interesting that, the thinking of HLA always being about self, non-self, kill it if it's an outsider, actually having all these other functions is something that really appeals to me. So that's the science bit done. For those of you who are not scientists, I just wanted to say, highlight all the collaborators and particularly the people at St George's and in the lab who've worked around this, and the funders as well. I'm not going to read through them all now, but a huge thank you to all of them. I said I would also talk not just about new roles and forms of immune proteins, but also for me and the roles that I've had at St George's, but also what those roles have meant in terms of that for other people. So, I'm trying to think, as I was at St George's, I'd been, as you take on some teaching responsibilities, I'd been a co-lead of a module. And then I think Fran's probably one of the people responsible here in that when Fran became the biomed course director, that left a year lead role vacant, which I took on, but also associate dean for research degrees. And when I was working in research degrees, around that time, we did a lot of work trying to improve outcomes for PhD students. And that was about implementing new regulations, training for supervisors, because I think a lot of us know you want PhD students to have submitted in 4 years. And at the time when I started in the role, we were about 55% at St George's for students getting through in 4 years. So there's a lot of work really about implementing ways of addressing that and trying to improve that. And we did. And at that time, that was under Tony Michael as head of the graduate school. But I think one of the big things for me was the Emerging Leaders Programme, which each was then division at St George's nominated some people to do, and that was led by Judith, who's there, who was chair of council at the time, and I think it was really your initiative, wasn't it, to set this up, and it was really about taking some of us and saying, okay, here you are now, but really what next, and how are you going to do it, and giving us a kick, and it was hugely influential for me, who was on it there, myself, Pippa, Fran, Judith Edison, Helen Mason, and during that, the role of head of the graduate school became available, and I kind of thought about that. But really through this program, I thought, okay, I'm going to apply for it. And as I said, now post-Coxing, if I go for it, I go for it, and I did. And so that took me on to being head of the graduate school, and in that time, partly through increased MRC funding, or largely through increased MRC funding to St George's, but also through our improved PhD submission and completion rates, we were able to apply with the London School of Hygiene and Tropical Medicine for this doctoral training partnership. And that was fantastic because it had been a while since St George's had been able to have MRC funded PhD students. So this was set up along the Northern Line with research priorities that were common to both St George's and London School of Hygiene and Tropical Medicine. And the bid writing was led by Eleanor Riley at LSHTM. And for me, that was like a masterclass in writing big grant applications. The way she went about it, the way we prepared, the way the writing went. And it was quite forward thinking or different thinking compared to other doctoral training partnerships, where often students come in a 4-year program, they go through a set first year, and then they go and do a PhD. Whereas our one was, students can do a master's first, but they can go straight into a 3.5-year PhD, which gives them a bit more time. But in that time, the students are selecting and directing their own training. They do a 3-month internship that's got nothing to do with their PhD. It can be with the WHO, it can be with finance companies, it can be whatever, but they do that. They choose from all the range at both institutions 3 modules that they'll take, so that's self-directed, but also there's flexible funding. If they want to do short courses, well, COVID now, but at a time around the world, specialised training, there's a pot of money, and they could pick short courses. I say they could, they still can, it's still going. And so it was a real student-directed program in terms of the training, and it's been interesting because there's been another round of funding from the MRC, and actually this student-directed aspect was something the MRC were particularly asking for, but which we'd already been doing for several years. So that took up a lot of my time and spent, meant I really was thinking more about student training and development as I went on through that. And that was something I was doing at St George's as well, through head of the graduate school and thinking of research degrees. And you can think a lot about training and the things that are involved, but I think a great learning experience for me was a suggestion that came from Ashton here, who I think might be listening online. And Ashton was a St George's student who'd come up with this idea for a book that wasn't the usual how to do a PhD book. It was one that had input from students as well as supervisors, because traditionally a lot of us think, depending on your generation, of PhD supervision is a bit like, I forgot the name of the scientist, but Beaker, and the other one is the supervisor just directing everything and knowing best. Whereas Ashton's idea was, we write this book, that have all the different aspects, what's important in year 1, year 2, prior to enrolling, and each chapter will be co-written by an academic, but also by a current or very recent PhD student. And he'd spoken to his personal tutor, Suman, about this, and Suman directed him to me, and we wrote the pitch, we got the commission, and it was everyone who was involved, well, the academics certainly, as well as the students, said the things they hadn't known, because it's all when I was doing my PhD, this is, but actually getting those two sides working together was sometimes frustrating, but always enlightening. And so it did change my thinking as well about PhD training and how that works. So if you want to buy it, you can get it on Amazon. It's a very short book, but very useful one with all those insights. But being head of the graduate school was not just about research degrees. It was about the top programs, the masters too. And certainly when I took it on, there was only 3 St George's Masters level programs that were available to people from outside St George's. And one of them, that third one there, on the left-hand side, had only just been introduced. And really there was a big move to increase the Masters provision at St George's to reflect all the things that we do and all the things that we're good at here. And so on the right is I think I checked it yesterday, what we have at St George's now. So it was a period of a lot of growth, a lot of course development, a lot of new course directors, and really thinking about what we do at St George's and translating that into courses that match our expertise, where we have real international profile. So it's very enjoyable to be overseeing that, and a lot of work as well. But some things that were coming out during that was this might not always be provision for people who'd come straight from undergraduate. Some of our provision, particularly around genomics, championed by the Kates, Kate Tatton-Brown, Kate Everett, was focused on, and in some cases HEE funded, for people who are healthcare professionals and upskilling them and developing their roles. And genomic medicine was designed by Health Education England specifically for that, to be part-time study for professionals working in the NHS. But Kate Tatton-Brown had also developed this course, which was for people training to be clinical geneticists, and also MOOCs as well. And so, we were thinking more and more about that lifelong learning, about developing people in their careers and perhaps opening up new doors to new roles or slightly different roles for them. And that led to thinking about professional education. This was, when Jane Safel came to George's, this was something she was particularly keen to establish. And this was taking it a stage further, not just about master's courses, but thinking more about short courses, online, flexible learning, distance learning. And so as head of the graduate school, I was involved in just setting up the structures for this to take us from not just our undergraduate and postgraduate degrees, which interface with our research, but filling that gap in between and thinking about professional development and professional education. And that's something that's ongoing, and we're still thinking along those lines about developing people throughout their careers and providing a range of courses to support that. So by this time, as head of the graduate school, I was involved in doctoral training, the things people do outside the lab or their actual PhD project, the training beyond that. I was now involved in things like short courses, I was involved in things like master's projects. But that came in line quite nicely with this NIHR funded big project, the collaboration. It's called Clark, I'm not going to read through it all, but it's about applied research. It's about applied research, and it was a big grant that was held by King's College London, St George's University of London, King's Health Partners, all the associated hospital trusts around there. And they had this, within that grant, a remit of capacity building. And what was capacity building? It's PhD students, master's students, short courses, training workshops. So that was quite a good fit for me. I came into it through a PhD summer school that had started with the Swan network quite some time before. I can't even remember how many years before, which was a network of Royal Holloway, Kingston, St George's, and was working to bring those closer together. That had then ended. We'd kept doing it. It then become adopted by Ms. Clark, and it was actually after running one of those summer schools I was asked to take on the lead for this area of activity for the NIHR grant. And so that was really doing the things I would do as head of the graduate school, but doing it across the NHS partners and the university partners of this collaboration. That grant came to an end, but NIHR then City College, they renamed it something much simpler, Applied Research Collaborations. And having led capacity building in the Clark for a while, I was invited to do the grant writing for capacity building in the new one. So that was 2 or 3 years ago now. So you can see all the things, all the different research themes in green that go on in applied research across this, and capacity building, which is cross-cutting across them all. So I wrote the capacity building section, submitted to NIHR. We got funding, so this is another 5-year grant. I think it's about 11 million across the 5 years. And capacity building actually has increased in its remit compared to the previous iteration. It's now not just researchers, it's about people working in health and social care too who are not researchers. So really what I spend some of my time now on is on this. So I lead the capacity building theme in this applied research collaboration, and we have a number of things that we do. So integrated training across the academic pathway, and that's led by Kirsty Coxon in the Faculty of Health, Social Care and Education, which looks after the NIHR fellows, but also supports people in applying for those fellowships. And we provide a range of things, short courses, scholarships and so on. Developing a module which will be available to St George's to use, as well as the partner universities that developed it, and various other workshops. One of the things that we were really keen to do when we wrote the grant, because NIHR had changed its academy structure, the support they provide to fellows, that got much more restricted in who they would allow to access their training events, and so came up with the idea of this effectively leadership academy for people who might not be able to access the full NIHR resources. And this is now going to be led by Scylla Harries in the Faculty of Health, Social Care and Education, who's recently joined the collaboration. And this is really about providing leadership training, action learning sets, workshops, mentorship. And we've just have a call out, I think it's recently closed, but we're looking for up to 100 people. They could be people in St George's Trust, in the Health Innovation Network, in St George's University of London, to access this training. And we'll be doing it every year. So the first year will start next year, which we're really looking forward to. And NIHR, we're quite keen on this, and I think we'll be interested to see how it works. I mentioned we've been given the remit as well of looking to provide a training and support for people who are working in the NHS and social care but are not researchers, with the aim of helping them implement research findings. So we have a whole strategy laid out for that, starting with local and developing a programme to thinking about rolling it out beyond London in the future. And we've had an initial run of that, which was done very much in partnership with the Health Innovation Network, which we'll be looking at feedback from just to see how we tweak it for rolling out beyond that. And so, in terms of my roles, I'm now, as of February 2020, Director of the Institute of Medical Biomedical Education. And we're a diverse group of people. I put in all the different sections, and there's a lot of us. And I have to say, having since I've been in the director, I love it. I work every day. I enjoy working with fantastic people, and I really look forward to us building our reputation. We have great work going on in MB. We have people working at the the interface with clinical research and helping develop guidelines and international guidelines. People like Hannah, people like Debu who do this work. We also lead nationally educationally. We've got people like Kevin Hayes, who's helping shape the upcoming, what is it, Hannah? medical licensing assessment, and also, you know, the prescribing skills assessment as well. Andrew Hitchens, you know, we are having an impact nationally on the field in clinical medicine and education, and you know, I think that, you know, it's been wonderful to do that, and we will keep building. There's fantastic research going on in the Centre for Biomedical Education as well. We have staff who are MRC funded, Wellcome funded. You know, there's a lot going on, and I think, you know, as I say, I work with great people and look forward to continuing to do that. But it's going to be changing, and I don't know if it'll be called the Institute of Medical and Biomedical Education anymore, because, you know, from next year, we're really looking forward to welcoming the Centre for Allied Health as well. And, you know, I said we do great work in NIMBY, but we've got a lot to learn as well from the Centre for Allied Health. We've got people like Julie Hendry and colleagues who are using their simulation and radiotherapy, and, you know, have industrial partnership for that, and will be doing fantastic work in that. And people like the paramedics and their Blue Light Champions scheme for students. I think it'll be fantastic. We're already integrating more in terms of our committees and how our courses run, and I think this will be a great thing to look forward to and see IMBY, although its name might change, develop as time goes on. And so I think, you know, that takes me to the end. You know, as I said, talked about new roles in the immune system, but as I said, you know, all those things like, you know, the training, the NHS, is something that is still very important to me and still want to continue to do. And I'd like to finish by saying thank you. And at one point, I thought of all the people I'd have to say thank you to, and I couldn't possibly put it on a slide. You know, everyone, you know, from Jenny, Deborah Bowman, all my, you know, previous managers, you know, Julian, Tony Michael, Paul Jones, all of them, you know, but also the people, the colleagues as well. You know, St George's is a fantastic place to work. There are so many people to thank. And so I just want to finish by saying thank you to everyone. Thank you.
Hello, Rachel. It is an absolute privilege to be asked to give the vote of thanks after your wonderful lecture. To say thank you for all of the things that you do for all of us, because a lot of what you do is for other people, and also to celebrate all your achievements, because you've had an amazing career so far. I'm sure it's still got a long way to run. I just wanted to touch on a couple of things. There are many people here, friends, family, colleagues, and we're all delighted to be here, and also online. So everyone is so pleased that we've come to this day, so thank you. Rachel talked about her research. I'm so glad that she did. Her research is so good, it's so important. She continues to be really research active and to mentor and support many people in the field. She mentioned Alex is here today, and I'm sure there's more online too. It's so important. A lot of what Rachel does has incredible impact, and I think sometimes we forget how important the impact is of the things she did. When she talked about the graduate school, for example, the MRC Lit is so important to St George's. It really underpins an awful lot of research here and boosts all of our teaching with all of these young people's lives who've completely changed because they've been able to access this program. And finally, Rachel is one of the most organised, thorough, busy people that I know, but she's always got time to support people and help people, and has a brilliant sense of humour. And I only wish I had more time to spend with her in the coffee shop or in the pub. And I'm sure many of us feel that way. So thank you so much, Rachel, for everything you do for all of us. And thank you for today as well. Thank you. And now it's time for the bar.
Yes.